Hi everyone, and thank you for joining us today. My name is Viantha Naidoo, and I am a microscopy specialist at the Africa Microscopy Initiative Imaging Center in the University of Cape Town in South Africa. And today it is my pleasure to give you a basic overview of Widefield and Confocal Microscopy, particularly focusing on the advantages and disadvantages of each imaging modality in order to help you decide which one to choose for your sample type. So let's start off with why should we use microscopy at all? And the answer is that microscopy provides spatial context. That is, microscopy helps tell us where certain things are and how they relate to where other things are. So in biology, to provide a few examples, this can mean it can tell us where or the subcellular localization of specific targets, such as mitochondria or lysosomes. It can tell us the organization of cellular components, the interaction between cells. It can help us study the interaction between proteins. It can tell us the location of specific cell types in a tissue. And it can tell us the changes in tissue architecture during disease and how that might further change when subjected to drug treatment. And these are just a few examples of many research questions that can be answered with microscopy. And so it really is a very useful tool. So we are going to focus on fluorescence microscopy today. And I asked the question, what is special about fluorescence microscopy? And the answer is contrast. So contrast is the ability to distinguish an object as separate from the background or other nearby objects. And so in fluorescence microscopy, you have fluorescence against a black background, which makes it very easy to identify structures and then analyze them. And to provide an example of contrast, here you have a triangle on the left that's light gray against the light gray background, and it's quite difficult to see. But when the triangle is darker, it becomes easier to see. And when it gets even darker still, it's much easier to see. And that is the advantage of fluorescence microscopy. So, as we mentioned, this talk is going to focus on Widefield and Confocal Microscopy. And I'm going to start off with some important questions that you need to ask when deciding on which imaging modality to choose. The first one is, what resolution is required? And here the resolution refers to the ability to distinguish objects that are separated in the actual sample as separate in the image. What is the sample type that you're working with? What is the size of the sample? And this includes thickness, which is very, very important. How fast do you need to image? What are you trying to measure? How would you like to analyze the data? And are the filters and light sources compatible with the fluorophores used? And these are just a few of many questions that you should be asking when deciding which imaging modality to choose, but these are some of the key questions. So let's start off with wide-field fluorescence microscopy. Here I have a few basic features of a wide-field fluorescence microscope highlighted, and we start with the light source. So the light will shine light of many wavelengths, potentially many wavelengths, and we have an excitation filter that will block out light of unwanted wavelengths. For example, if we were only interested in blue light, as is shown here, all the other wavelengths of light will be eliminated. So the blue light goes to a special type of mirror called a dichroic mirror, which reflects light of certain wavelengths and transmits light of other wavelengths. So the dichroic mirror will reflect the light to the objective and to our sample, which in this example includes fluorophores, so fluorescent molecules that have the potential to be excited by blue light. And when they are excited by blue light, they will emit green light. And then the green light goes through the objective, through the dichroic. So now the dichroic is designed to let through green light to the emission filter, which will block out any additional unwanted wavelengths of light, to the detector, which is a camera in a wide-field microscope. So importantly, light from the focal plane is going to the detector, but so is light from molecules that are below the focal plane and above the focal plane. So all light emitted from all these molecules are going to the detector. So some key takeaways of wide field fluorescence microscopy: the whole sample is illuminated using LEDs or lamps. Large fields can be imaged. It is fast, it is sensitive, which means you can get bright signal even for weak fluorophores, relatively easy to work with. But there is a drawback, and that is that out of focus light is seen in the final image. Remember, light emitted from molecules above the focal plane and below the focal plane are going to the detector. So for thin samples, there'll be less out of focus light, but for thick samples, you'll get lots of light scattering, which can lead to a blurry image. So in order to better see the structures in such images, you will need additional computational techniques. And that would mean, which is deconvolution. So here's an example where you have a wide field fluorescence micrograph on the left, which you can see is very blurry. And then when you apply deconvolution methods to that image, you start to see structures much more clearly. And this is very promising. So, however, this requires a lot of expertise. You need to use good models of deconvolution. You need computers that are very powerful because it can be computationally very intensive. And it can lead to artifacts, especially if you don't know how to properly use deconvolution. So how else can you get rid of out of focus light? And the answer is confocal microscopy. So confocal microscopy uses this technique called optical sectioning, where essentially it's using a pinhole to reduce the out of focus light. So it is analogous to physical sectioning, where you would cut sections, thin sections, and then you would stain them, and then you would image them on a microscope. Except with confocal microscopy, you can stain a thick sample, and then you can image that and let the machine do the sectioning for you optically. And here's an example of a Drosophila embryo, where I'll play the video for you. And here it's going through the thickness of the sample. And you can see that you're getting different amount of detail in the different sections. So this really is equivalent to what you would have as physical sections if you did normal sectioning and staining. Except here, the machine is doing it for you. So let's go to the basic light path of a confocal microscope and see how that compares to a wide field microscope. So again, we start with the light source, but here the light source is a laser, and that helps narrow down the wavelength that is used for the excitation. So that light goes through the excitation filter, which blocks out any wavelengths we don't want, again goes to the dichroic, through the objective, and to our sample, very similar to wide field. Again, molecules in all the different planes are excited, and they emit light, which then goes through the dichroic, through the emission filter, but now we have a special thing called the pinhole aperture, which is going to let in light only in the focal plane. Light emitted only in the focal plane to the detector. So here, the molecule in the focal plane, its light is going through the pinhole to the detector, but the molecule below the focal plane, that light is blocked. The molecule above the focal plane, the light it emits is also blocked. So that results in a much crisper, high contrast image. So if we have some key takeaways for confocal microscopy, sample is illuminated in a small area using lasers. The traditional confocal microscope uses point scanning. There are no cameras. It uses other detectors that amplify signals. And in this case, we have a photomultiplier tube. And then the key feature, which I've mentioned, is this pinhole aperture, which filters the out of focus light. And the result is improved contrast and enhanced practical resolution, which means you're able to more easily tell two things apart. The drawback of a traditional confocal microscope is that the imaging is slower because of the point scanning, and you can get increased photobleaching and phototoxicity if the acquisition settings are not optimized, and that is either damage to your fluorophores or damage to your sample, and you can get limited penetration depth in thick samples. So just to show you again the comparison between the wide-field microscope and the confocal microscope, the light sources are different. And importantly, the confocal microscope has this pinhole aperture which blocks out out-of-focus light. And then the detectors are different as well. So here I would like to show you an example of a wide field fluorescence image versus a confocal image of the same area. And you'll notice that this image on the left is a lot brighter, but it is blurry. And that is because that out of focus light is in that image. Whereas in the confocal, this is a single optical section, which means that out of focus light has been blocked by the pinhole. So both images can be useful depending on what you would like to analyze. For example, the image on the left might still be good for counting the number of cells, for example, even though it's blurry. But if you needed to understand further detail, you might need to use the image on the right, which is a confocal image. And I just want to showcase that wide field does not always result in very blurry images, particularly for thin samples. Here is an image on the left, which is of a mouse brain tissue section. And you can see there's very good clarity. And this is an image of cancer cells, and it looks very, very crisp, very much like a confocal image, but it is actually a wide field image. So this leads me on to the triangle of compromise. So in imaging, you are often trying to optimize three factors, and that is sensitivity, resolution, and speed. And you can't fully optimize all three factors at the same time. And you have to balance these factors depending on the goals of your imaging experiment. For example, if you need more detail, that is resolution, like if you want to understand subcellular detail, then you will need to favor resolution over speed and sensitivity. But if you're working with weak fluorophores, for example, then you would need to detect as much signal as you can, so you're favoring sensitivity. And if you're working with live cells that are very sensitive, you might want to do a very fast acquisition, so you might favor speed over resolution and sensitivity. So the best signal to noise ratio, which is essentially your signal above your background, is often a balance of all three factors. So just to summarize some important information of wide field versus confocal microscopy, and that is for your sample thickness, for wide field microscopy, you're working with very thin samples in order to give you good images. So ideally, you want to work with samples that are under 5 microns thick. For confocal, this can be 5 to 100 microns because of the optical sectioning. Your sample illumination is full field for wide field, but for a traditional confocal microscope, it is point. The depth of field is large for a wide field, but can include out-of-focus information, and confocal it's small but high contrast. The resolution is lower in a wide field, and again, resolution is the ability to tell two things apart, the resolution we're speaking of in this talk. Whereas in confocal, it can be higher because the optical sectioning improves the contrast. And then the sensitivity is higher in a wide field microscope than a confocal. The speed is fast for wide fields and slow in a traditional confocal, which uses point scanning. The complexity is simpler in a wide field compared to a confocal, and the cost is much more affordable for a wide field versus a confocal. So a final concluding statements for each imaging modality is a wide field is preferable for fast or large scale imaging, so you can get good practical resolution, that is, the ability to tell two things apart for thin specimens, whereas confocal is optimal for imaging thick or 3D samples. And then this principle of optical sectioning enhances the contrast, enabling identification of separate objects. Lastly, I would like to acknowledge a number of people who have supported my microscopy learning journey and also or have also provided advice for this talk. And those are doctors Michael Reike, Anya Schmidt, Chad Hobson, Teng Leong Chu, Paula Montero Leopez, and Jennifer Waters. And lastly, we are very grateful to the funders of the Africa Microscopy Initiative, and that is the Bill and Melinda Gates Foundation and the Chan Zuckerberg Initiative. And we are also grateful to the University of Cape Town, and particularly the Institute of Infectious Disease and Molecular Medicine, where the Africa Microscopy Initiative Imaging Center is hosted. And thank you all for joining today. I hope this talk was useful. And if you have any questions, please feel free to contact me using the information on the title slide, and I will be happy to chat further. Thank you again.